Now to proteins. We're going to talk more about proteins and nucleic acids than the other two kinds of organic molecules all year. And they have much more to learn and much more variety. So here we get started, okay? So here's your EOC thing. Describe the structure of proteins and amino acids. They separated those two because they're not really synonyms. A single amino acid, like you see a bunch of here, these are all single amino acids, are monomers. They're the monomers that when you put them together, they make up the polymers that we call proteins. But just technically, an amino acid is not a protein. It's not the worst thing in the world to say that. If you had to identify it, you certainly wouldn't identify it as a carbohydrate or a lipid or a nucleic acid. If that's your only other choice, you would call it protein. But technically, an amino acid is not yet a protein until you stick it to at least one other ones. Okay, so explain the functions. We'll get to that. Identify some reactions amino acids undergo. You know what? They're hinting at those reactions they told you they're not going to ask you about. We'll see that on the next slide. Dehydration, synthesis, and that's the only reactions I can think of that amino acids take part in that they would be hinting at there. So anyway, look at each of these. You're not going to have to memorize these. But given any one of them, you need to be able to identify that this is not a carbohydrate, this is not a lipid, that this is a protein kind of thing that we call an amino acid. And if several are stuck together, you would need to be able to identify that. We'll see that picture in just a minute. So what identifies them as these amino acids? Well, if you look at what's highlighted in white, you can see in every one of these it's different. That's the part of each amino acid that makes each one different from the other. Each of those groups of atoms is referred to in general as an R group. That's just the alphabet letter that somebody chose long ago to identify this group that every amino acid has. So now look at the rest of it. What else does every amino acid have? It has a carbon that R group is stuck to. Now here's something I want you to notice. If we take this picture, for instance, and we turn it upside down so that this, the simplest of R groups, is just a hydrogen atom, right? If it's at the top, or if this CH3 was at the top, it's still an amino acid. If you turn a molecule upside down, you haven't changed it. So you might see these with the R groups on the side and all these other groups in a different place, but they're all still going to be the same. There's a carbon in the middle. It's attached to an R group, and it's attached to a simple hydrogen. You notice all of these have one hydrogen attached to that central carbon. And then they all have two things which give these the name amino acid. One of them is an amine group. That's a nitrogen. That means all proteins have nitrogen. And then that nitrogen is attached to two hydrogens. Now notice up here, that doesn't look quite like what I drew. That's NH3, 3 hydrogens. Well, here's the difference, because you might see either one on a test. You might see either one. It's kind of like the glucose thing I showed you. This is what it would look like if it was dry. But if you put it in water, this nitrogen is good at stealing protons, which is like a hydrogen ion, right? And that's why you see 3 hydrogens here and a little plus sign. I know that's kind of small for you to see, right? So this is called an amine group, and I've drawn it when it's dry. This is what it would look like if it was wet, if this was all in water, which it always is in a living kind of organism. But still, it's called an amine group. That's why this is an amino acid. And on this end is that group that I identified as a carboxyl group real fast, right? I identified that because it was on a fatty acid, this carboxyl group. This is what makes it a fatty acid and what makes this an amino acid. What's an acid? Again, an acid is a substance that when you put it in water, it loses a proton. And what does that proton do? It gets grabbed by water to make a hydronium. And remember, the more hydroniums there are, H3O plus, the more hydroniums there are, the more acidic that thing is, and the lower, remember, the lower its pH number is. Well, when you put this thing in water, the nitrogen part grabs a proton. That makes it a little basic. And the carboxyl part loses, not this whole hydrogen, but just the proton part. That's what leaves it negative. And again, these pictures are all what amino acids look like if they're in water. So I drew it here dry. You might see this sketch on a test. You might see one of these sketches. You might see the symbol R. You might see a specific kind of group, but you're expected to be able to recognize, oh, look at that. There's a carbon in the middle. It's got an amine group stuck to it. It's got a carboxyl. It's got a hydrogen. This must be an amino acid. It can't be anything else. There's no lipid that looks like that. There's no carbohydrate. There's no nucleic acid. Okay? But you notice these R groups can look quite different. They can have like carbohydrate rings stuck to them or other kind of things. They can also have different elements like sulfur. So here's another aspect about proteins. Proteins can, and most of them do, have sulfur in them. Only a few of the amino acids have sulfur, but your average protein is made of hundreds of amino acids, and so it's likely they're going to have sulfur. Here's another thing about proteins. They don't have phosphorus in them. If you see phosphorus, as you already saw, it might be a phospholipid, or as we're going to get to it, it might be a nucleic acid. All of them have phosphorus, but it is not a protein if it has phosphorus. Likewise, if it has sulfur, it's not a nucleic acid, and it's almost certainly not either a carbohydrate or a lipid. So sulfur becomes something that really identifies proteins. Nitrogen identifies proteins, but nitrogen is also, as we're going to see, found in all nucleic acids. So if it has nitrogen in it, that doesn't mean for sure it's a protein. Okay, on to some more complexity. Here's a familiar picture, right? You have a little molecule, and you have another little molecule, and we are sticking one to the other. This again is dehydration synthesis. How would we get them apart? Same idea in reverse, hydrolysis. We would take a water molecule and split it, and part of it would end up here, part here. For amino acids being attached to each other, notice that the water always comes from the carboxyl OH is going to come off, and one of the H's of an amine, and that's where the carboxyl carbon is going to join to an amine nitrogen. And for no especially good reason, somebody decided to give that kind of covalent bond, because that's what it is, 2 electrons being shared there, right, symbolized by a solid line, decided to give it a special name of peptide bond. And that's why if we have many amino acids stuck together, which most proteins are by far, hundreds or even thousands, we also call that chain a polypeptide, referring to the peptide bonds here that there are many of, as opposed to the amino acids. Okay, so as you can see, all of the organic molecules, we've talked about 3 types so far, and the nucleic acids are the same. They get put together by dehydration synthesis. They get pulled apart by hydrolysis. So now, compared to the others, proteins show a great amount of variety. A great amount of variety in types. You have several thousand kind of proteins making up you, but only several hundreds kinds of carbohydrates and lipids. And what is different about each protein is the type of amino acid that's stuck together, but also the sequence. There are about 20 different amino acids. But if you had, for instance, 20 different colored beads and you were asked to make a necklace and you had multiple amounts of each of those 20 different colors, you could technically make an infinite number of different necklaces by putting different colors together in different sequences and in different lengths. This one has 20, this one has 1,000, so on and so forth. So sequence is going to be a very key word here, especially as we go on later and talk about nucleic acids. So let's talk about some protein functions, okay? As you might imagine, if there are so many more different kinds of proteins than carbs and lipids, they probably also have so many different kinds of functions than they do. All right, let's look at two functions at first here that they have in common with carbs and lipids. One is many protein molecules form the structural parts of cells and therefore organisms, such as the keratin that makes up a lot of your hair and nails, the collagen that makes up a lot of your skin, the silk that makes up spider webs, and a whole bunch of other structural proteins, just like cellulose is a structural carbohydrate and phospholipids are a structural lipid. Lots and lots of proteins have a structural function. Also, unlike any carbohydrates, but like those lipid hormones that we called sterols, testosterone and estrogen, many proteins function as messenger molecules. Insulin tells your cells messages about blood sugar, human growth hormone, obviously growth messages, which means cells, we want you to divide, basically, and a whole bunch of other. In fact, there's many more protein hormones than there are those sterol lipid hormones. This is a kind of protein and function that we're going to spend a whole unit on, enzymes. That's the name of the proteins that have the function of being a catalyst, which means they slow down, they can slow down, but most of the time they are speeding up chemical reactions. There's all different kinds of catalysts, like heat, but the kind of catalysts that are protein molecules are called enzymes. Their names often, not always, but often have an ASE ending, not OSE, like those carbohydrates. Lots of confusing vocab here. My condolences, right? So for instance, the enzyme that helps you break down table sugar into glucose and fructose is sucrase because it helps break down sucrose. At least that makes sense. Then there's a bunch of proteins involved in things moving from one place to another. Some of them are involved in things moving into and out of cells. They're in the cell membrane. And then some of them are involved in moving things all around in an organism, like those in your circulatory system that include hemoglobin. Hemoglobin picks up oxygen, carries it around, drops it off, picks it up, drops it off. It's like a little delivery truck. It's a protein. We are going to see hemoglobin a bunch of other times, including coming up real soon. Many proteins have a defense function, and you should have heard about these by now, for sure. Those proteins are called antibodies. They are made by certain white blood cells in your immune system, and they very specifically stick to things because of their very specific shape. And those we're going to spend a lot of time on in a different unit later on in the year also. There's other functions that proteins have, but I just listed these four, or five rather. These are the big five, and especially I would say these enzymes where we're going to have a whole unit. Okay, so like I said before, the function of any kind of molecule has to do with its structure. A lot of its structure includes its shape, also where it might have some positive and negative charges, right? But here, especially when we get back to enzymes, we are going to see what happens if a molecule loses its shape, its natural working shape. Therefore, we call it denatured at that point. And that means it's not going to work because we're going to see that these molecules are very much like puzzle pieces. They have to be the right shape to fit into another molecule and do what they do. And if they are denatured, that shape is changed, they're not going to work.